Thank you so much, Dr. Buttermore, for that really wonderful and kind introduction. And thank you to the entire data science team and the neuro-oncology team for inviting us here today. I'm excited to share with you a bit of an introduction to the work that will be presented today for the paper at hand. And then I will turn it over after that to Patricia Nano, who is really the better person to talk about this work because she's the first author and the one who really got into the weeds. And so she'll be able to answer a lot of questions, and I will be here too. But when thinking about the developing human brain, I always like to start with this image. For those of you who aren't familiar, this is a picture of a gestational week 6 human brain. And as you can see, it is structurally very different from what you would eventually imagine to be the human brain that is fully developed and adult. And when we think about the human brain, my lab is particularly interested in studying this outermost wrinkly layer, which is the cerebral cortex. The cerebral cortex in the human gives us a lot of really important capabilities and functions, cognitively being able to integrate a lot of sensory information, look at decision making, engage in judgment, etc. And for the longest time, thinking about the cortical areas that exist, each colored here by a different hue, you can see that although each of these layers of the cortex, which is typically a 6-layered neuronal structure, are preserved across the cortex, there are important differences across the different spans of the cerebral cortex. And these drawings have observed this for more than 100 years. What we are also learning, though, is in addition to morphological differences across the gradients of the cortex, there are important functional differences in terms of their transcriptome, electrophysiology, connectivity, morphology, etc. And importantly, the vast majority of the blueprint to develop these areas is established pre-birth because the vast majority of stem cells that exist in the human cortex are not present after birth. Therefore, if we want to think about questions such as neurodevelopmental disorders, psychiatric disorders, and the vulnerability towards these, it's really important to study these developmental time points. It's also essential to think about this in the context of responding to injury because stem cells are really a developmental cell type, as well as when thinking about neurodegenerative diseases. We won't highlight it here, but we are also observing that many of these cell types and trajectories are reactivated in the context of brain cancer. And so understanding the developmental underpinnings and their trajectories are really essential to being able to understand these devastating cancers. Again, for decades, from the work from Ramon y Cajal as well as Wilhelm Hess, who were some of the first scientists to observe and draw the cell types in the brain, it became very clear that there were important differences between the adult brain and the developing brain. And we can see this through the morphology. And more recently, with the advent of single-cell RNA sequencing, it's become clear that not only are these differences morphological, but also there are important differences in terms of the single-cell transcriptome and the evolution and maturation of these cells towards their adult state. One of the most comprehensive first studies to look at this was only 4,000 cells, but gave us really important insights in a work led by Tom Nowakowski in 2017, where profiling about 48 individuals allowed us to explore some of the key trajectories as well as areal distinctions across the developing human brain. And single-cell RNA sequencing since then has only expanded our reach in terms of understanding the cell types and trajectories that give rise to the human brain. Now, for those of you who are not cortical development aficionados, I'll give you a quick overview of many of the cell types that exist during cortical development. What you saw in the beginning was largely comprised of neuroepithelia, and for the most part, we believe that the neuroepithelial sheet that gives rise to much of the cortex is largely uniform. This then gives rise to radial glia, which are able to subdivide into a number of subtypes over developmental time, giving rise to neurons either directly or subtypes of radial glia, including truncated or outer radial glia. These then give rise to intermediate progenitor cells, which give birth to newborn neurons that migrate up the radial glia scaffolding in order to first generate the deep layer populations of the cortex, followed by the upper layers of the cortex. When gliogenesis is mostly complete, When neurogenesis is mostly complete, these radial glia then switch to a process called gliogenesis, making glial populations in the cortex, such as oligodendrocyte precursor cells and astrocytes. Now, one thing I always like to highlight is when thinking about the human developing brain, each of these populations in red are ones where we know that there is either enrichment, specialization, or unique subtypes that exist in the human brain compared to rodents. Meaning that if we really want to think about the processes of human development and how they generate vulnerability to human neurological disorders, we also need to study human cells. In previous work that I performed in collaboration with Dr. Arnold Kriegstein and Dr. Tom Nowakowski during my postdoc at UCSF, we were really interested in starting to generate some of these developing human cortex cell atlases by taking samples from before the second trimester and performing anatomical dissections followed by single-cell RNA sequencing. And from this, we were able to ask what cell types and subtypes exist in the developing brain, how are they distributed, and how do they change over time. And from this, we were able to generate an atlas shown here of 700,000 post-quality control cells. I think most people here will be familiar, but each dot is an individual cell colored by the major cell type, which was inferred from our analysis. But we have many other metadata properties, such as the age of the sample or an anatomical dissection. And this atlas is from peak stages of neurogenesis, shown here, from the second trimester across 11 individuals. And what we've been able to do with this data is to generate a public data resource of cell types and trajectories from the UCSC cell browser. If you're interested in this data or the data that Patricia is going to talk about, please do visit this site. There's a really great repository of data sets there, and you can interact with them without bioinformatic expertise. So if you want to see what your favorite gene of interest is, just go check it out, and it's super easy to do. And if you want to play with the data, you can also download it. We've also used these data to compare to in vitro organoid models and to benchmark the strengths and weaknesses of these in vitro systems, which are really crucial for studying the functionality of human cell types. And although we won't talk about it, compared to the development of brain tumors. Overall, my lab is really interested in understanding how neural stem cell fate specification occurs to give rise to the diversity of cell types that exist in the adult human brain, but also how they're reactivated in the context of glioblastoma. As much as possible, we try to anchor ourselves in primary samples and primary data sets because we do believe that ground truth really matters. But when we need to perform manipulations that are not feasible in those systems, we do lean on cortical organoids, which are a stem cell-derived system that you'll hear about today as a way to test hypotheses and perform and identify new insights. We perform a number of measurements across these axes of variation, including related to cell type heterogeneity, gene expression, and metabolomics, which we won't highlight today, but if you are interested, please do follow up. So overall, the overarching questions we're asking are, how do human neural stem cells build the human brain, and why are they so devastating in brain cancer? Now, before I turn it over to Patricia, I'd just like to make a quick plug that we are constantly expanding and improving the atlases. And in collaboration with 5 institutions, we are in the process of the next stage of the BRAIN Initiative, called the BRAIN Initiative Cell Atlas Network, or BICAN, where we are able to sample 30 brain regions this time across the brain, across 4 developmental time points, and looking at human, macaque, and marmoset in order to also think about the evolutionary specializations of human. So if you're interested in this data, it is released quarterly, and we're working on putting it together with analysis and annotation, which will also be available on the browser when that's ready. And Patricia's actually leading that work as well. So with that, I will turn it over to Patricia and look forward to her sharing some of the exciting work that's happening. I will highlight that Patricia is not only an exceptionally talented postdoc who innovated the methods and both experimental and computational here, but she's also thinking about how to apply this to neurodevelopmental disorders in her own group. So if you or anyone is hiring, please do keep that in mind for faculty positions. And with that, take it.
All right, thank you so much, Aparna, and thanks to all of you for giving me the chance to speak more about our paper. Very excited, and then as Aparna alluded to earlier, this paper really centers around the question of how does the human cerebral cortex get this vast diversity of cell types and this organization of functionally specialized cortical areas. that give us the ability to think and feel. Now, it is, of course, these processes that make these different types of cells and these different unique areas that is often disrupted in the case of neurodevelopmental disorders and other diseases, as Aparna alluded to earlier. But our ability to understand all of these neurological conditions is really challenged because we don't have a very clear molecular understanding of how the human cortex forms to begin with. So I spent the better part of my postdoc trying to generate more comprehensive molecular profiles of the human cortex, particularly throughout its development, extract mechanistic hypotheses from these profiles, and then test these hypotheses in vitro models of the developing human brain. Now, this paper really builds on recent advances in single-cell profiling, and that, along with work from consortiums like NIH Brain Initiative, have given us now these vast data sets, particularly successful for the mouse. So, for example, we have these large data sets profiling the developing mouse cortex, and even more recently, we have data sets now of the entire adult mouse brain. But as Aparna alluded to earlier, there are many human-specific aspects of cortical development. And so there's been an equally important push towards understanding human cortical development. And many of these data sets now have given us a picture of the adult human cortex and many different adult cortical areas. And as we've seen before, there are seminal works looking at specific cortical areas at certain time points of human cortical development, including a recent study looking at the second trimester human cortex all the way through adulthood through 8 time points. However, because of the relative inaccessibility of developing human primary cortical tissue, we still don't have one profile looking at the entire human cortex throughout the entirety of the lifespan. And so what I asked is, can we develop a computational framework to take advantage of all of these different data sets that already exist? And towards that goal, we generated a meta-atlas, first of the developing human cortex. Now, this meta-atlas contains 7 of the recently published data sets that I just showed you. That's 96 individuals and almost 600,000 cells spanning peak stages of neurogenesis. Using conventional batch integration approaches, we see in this UMAP of the cells in our meta-atlas that our meta-atlas still contains the cell types that one would expect in a human cortex. We have our excitatory neurons and our radial glia progenitor cells. But then, when it came time to ask how can we use this atlas to extract mechanistic hypotheses of how these cell types are formed to begin with, we had to keep in mind how do we dampen the technical variation between these different atlases or different data sets without also dampening the very subtle but real biological difference between different cell types that has been described during development. And even further, how do we extract gene networks from these atlases that give us mechanisms of how these cell types are formed? So towards that goal, we also generated a novel gene co-expression analysis pipeline to create what we call metamodules. Now, to create a metamodule, you have to go back to each of your 96 individuals, in this case, in our meta-atlas. And for each of these samples, I conducted hierarchical clustering of the cells based on transcriptomic similarity. So that clustering will give us clusters, they have cluster markers, and so those individual level cluster markers represent the biological variation within each sample. We have a gene score metric that allows us to ask which of these cluster markers are the best representative of their home cluster. And so we pool all of the very best individual level cluster markers together, hierarchically cluster those to generate metamodules. And big picture, these metamodules represent individual level signatures that together throughout the entire meta-atlas travel in similar patterns. Now, to make sure these metamodules don't represent just a random assortment of genes, I went through each of the 225 metamodules generated by our pipeline, and then, using GoTerm enrichment analysis and literature review, functionally annotated these modules, which I was able to assign some sort of biological process to for the majority of our modules. You'll see in this list, we have some cell type specific modules, or at least modules associated to some sort of cell type, like astrocytes or maybe synapses. We also have some modules that represent a more broad category, for example, cell stress or metabolism. And what's nice is that these cell type specific modules give us an ability, or I should say cell type associated modules, give us an ability to have a sanity check. And you could probably predict what I'm going to show you next. We then ask if these cell type associated modules are indeed linked to the appropriate cell type. So for example, vascular function modules are most active and most enriched in endothelial cells. Almost or like half of our microglia associated modules are associated with microglia. And we also have glia-specific or glia-associated modules. We don't see as strong of a enrichment in glial cells in our meta-atlas, but again, our meta-atlas spans peak stages of neurogenesis. It might be simply too early to see a very strong signal from glial cells in our data sets. Okay, all of this gives us greater confidence that what we're seeing here are meta-modules that can give us insight into biological processes of how cell types are initiated and refined and through what mechanisms. But before we really started going deep with these tools, we had to ask ourselves first, is it better to take developmental modules found in a developmental context and then track them throughout the lifespan? Or should we actually instead go backwards and ask what are the meta-modules in the adult cortex and how do they map back to the developing brain? Okay, in order to really answer this question and address it, we had to make, again, another meta-atlas. So this is a meta-atlas of the adult human cortex. It's 16 data sets, 274 individuals, and almost 3 million cells. So our pipeline can scale up. Now, to really ask the question of how do developmental metamodules and adult metamodules compare between these different contexts, we created a tool that allows us to ask this question systematically. So this tool is our module specificity score. And this is simply a score of, for each module, how specific and enriched it is in each different subtype. And so what you're seeing here are all of our developmental modules. And I'm asking how specific and enriched are they to specific or to different developmental subtypes. And that score here is represented in blue. Big picture, when I saw this heat map, I saw a lot of blue. And what that means is that developmental subtypes seem to be associated with groups or sets of specific modules. And this is in line with what the field has shown, which is that during development, the distinctions between different subtypes are very subtle. And then subtypes exist along a continuum. Okay, so let's ask the same question in the adult. Now in this heat map, I have my adult modules, and I'm asking how specific and enriched are they in adult subtypes. Again, that score is represented in blue, much less blue in this heat map. Again, what that means is that these adult subtypes are now here characterized by just one or two specific modules. And because the score is so systematic, you can then play mix match and now ask how do developmental modules play in the adult, how do adult modules play in development. Either way, the trend still holds. Developmental subtypes are characterized by a greater number of specific modules. Okay. Now that we have this tool and now that we've interacted with it a bit, we can now ask our question of how does the cell type specificity of these different modules change from development to adulthood? And what I'm about to show are two Sankey plots. For each Sankey plot, I'm going to show you the modules that are specific to, in this case, adult subtypes. Where are they specific in the developing human cortex? And then vice versa here. What I expected to see here was something a little bit more straightforward. For example, if I have modules that are specific to adult oligodendrocytes, they would then be specific to oligodendrocytes during development. Reality is a little bit more complex. And what this showed me, big picture, is that the cell type specificity of these modules diverge and change from development to adulthood. So in our eyes, the most straightforward way of using these modules to track how the human cortex forms is by first looking at how the modules act in the developing human cortex and then track them throughout the progression of the lifespan. So now we are able to now ask questions of how do these cell types form, get refined, and how and what mechanistic ways. And what I'd like to do is show two vignettes from our paper where we interact with our modules in this way. The first is a module that's called Module 156. And by annotating these different modules, I found that it was related to radioglial biology. And in fact, in our developmental meta-atlas, Module 156 is highly active in our radioglial progenitor cells. And throughout development, Module 156 activity does trend upward. In the adult, Module 156 is still active, as you can see here in a recent adult cortical dataset. But interestingly, it's very specific to glial cells. And so we asked, well, maybe one module 156 might be signaling somehow a shift from neurogenesis to gliogenesis, like in the developmental stages that we talked about much earlier in this talk. Okay, now if that was true, then we should be able to see module 156 members in the ventricular zone that's rich with progenitor cells in the developing human cortex. And so we were able to get human primary cortical tissues from gestational weeks 16 and 20. Those cortical slices are shown here. And we stained them for two module 156 member genes. One is QKI, a transcription factor that's already been linked to glia biology. It's a nice positive control. The other is PIDLM5. It's a staphyloid protein that has not been linked to glia before. And so if we zoom in to the progenitor-rich ventricular zone here, what we found in these developing human primary cortical tissues is that QKI-positive nuclei in green are surrounded by PIDLM5 punctate staining, lending support that this module exists in progenitor cells and may play a role in switching those cells from neurogenesis to gliogenesis. Now, while we weren't able to test the mechanisms of that specific function here, we did conduct such a targeted mechanistic analysis for a different module, now looking at how deep layer neuronal functions might be specified. So this module is now called Module 20. In our meta-atlas, Module 20 is most active in excitatory neurons and particularly deep layer excitatory neurons, where it has this early and sustained activity throughout, or increase in activity, I should say, throughout development, and you're not seeing that in upper layer neurons. Module 20 is still active in the adult. It's still active in deep layer, that is layer 5, layer 6 cells. But interestingly, Module 20 activity is highest in deep layer neuronal subtypes linked and elevated with FESF2. FESF2, of course, is a transcription factor that the field has shown to really drive deep layer fates. That's exciting and everything, but FESF2 is actually not in Module 20. In fact, it's not even expressed at the same time as Module 20. Instead, in our developmental meta-atlas in deep layer neurons, FESF2 actually increases or peaks in activity here in green before the peak of Module 20 activity here in purple. However, half of Module 20 genes are indeed putative targets of the FESF2 transcription factor, including the only transcription factor in this module, TSHC3, which you can see here in green peaking in activity between the peak of Module 20 and FESF2 levels. Okay. Now, all of these different expression patterns, they're not actually conserved in the developing mouse brain. And so if we wanted to ask, how do FESF2, TSHC3, and Module 20 drive the specification of deep layer fates in the human brain, we need to have a human experimental context. And this is that cortical organoid model that we alluded to earlier. So as a review, for those who are unfamiliar, organoids are derived from stem cells, and then by pushing them through phases of cortical patterning, differentiation, and maturation, we can get these little spheres. And in these cortical organoid spheres are rosettes. Rosettes are aligned with progenitor cells and mature neurons that mimic the cell types that we see in developing human primary cortical tissue. And this architecture of progenitors in rosettes lined with mature neurons mimics the progression of these cell types in the developing human primary cortex. Okay. Now, much work has been done to really expand and improve upon the organoid model. And one thing that we were really excited to see is recent work from Paula Arlada's lab, taking these organoid models and now generating chimeroids. In the chimeroid model, they generate multiple brain cortical organoids from different donors, and then through a process of dissociation and reaggregation, can put these donor cortical organoids all together in the same mass, enabling parallel investigation of multiple genetic backgrounds. That's already very exciting. But one part here that really piqued our interest is that the step of dissociation and reaggregation, that's actually a great way to introduce genetic perturbations into this organoid system. And so with this model, I set out to test are FESF2 and TSHC3 and Module 20 required to drive deep layer fates. So I generated organoids from 3 different stem cell lines, and then I first dissociated and then reaggregated these cortical organoids together in the presence of an shRNA. And that allows me to knock down FESF2 and TSHE3 either alone or in a double knockdown, grow the organoids for 8 weeks, and then use flow cytometry and single cell profiling to ask in these perturbed cells, what are the effects on cell type specification? Okay. So as our computational predictions suggested, the knockdown of FESF2 and TSHE3 either alone or in a double knockdown, that was sufficient to decrease Module 20 activity. And what was particularly interesting is that in our double knockdown, we see a decrease in deep layer neurons that is accompanied by an increase in radioglia progenitor cells. We were able to validate this using immunofluorescence staining of radioglia markers in our perturbed cortical organoids. So to get at this mechanism a little bit more deeply, we then looked at the effects of each of these perturbations on all of the different Module 20 genes. And what we found here is that the double knockdown and knockdown of FESF2 decreased the gene expression of most of these Module 20 genes. But that's gene expression. When we actually looked at chromatin accessibility, we find that now it's TSHE3, it's not FESF2, that is required for chromatin accessibility of FESF2, TSHE3, and most of these Module 20 genes. And so, taking all of this together from our computational predictions all the way through our experimental approaches, we have this mechanistic model wherein waves of FESF2 and TSHE3 acting on different modes activate Module 20 and permit the specification of deep layer fates. And so big picture, what we've been able to do with our meta-atlas and meta-module approaches is make sense and take stock of all of these different cortical data sets. And now we've been able to collect them together into two very interactable resources, the developmental and adult human cortical meta-atlas, both of which are in the UCSC cell browser for the field to use. In addition, for those who want to use our meta-atlas and meta-module pipelines to ask their own biological questions in a different system, we made that pipeline available in our GitHub as well. Okay, with the time we have left, I'd love to share a quick peek about how I've been using these meta-atlas and meta-module tools to expand upon them and go into further research directions. One thing that we've been doing with these meta-atlas strategies, as Aparna has mentioned earlier, is to try to make bigger and more expansive data sets. Because of course, these meta-atlases, they're only as good as the data sets used as input. So for example, we are using these meta-atlas and meta-module approaches as part of the new generation of NIH Brain Initiative projects, particularly with our collaboration with labs at UCSF, Yale, and UW-Madison, where we're using these approaches to look at 3 different species, human and 2 non-human primate, 4 developmental stages, 30 brain regions. And I'm particularly excited here because these brain regions include 14 cortical areas that have not been very extensively profiled before. In addition, I'm co-leading a joint analysis working group within the NIH Brain Initiative, where we're taking these meta-atlas approaches as well as other types of integrated analysis approaches to pull together existing and leveraging to be generated NIH Brain Initiative data sets to create other meta-atlases, maybe of just the prefrontal cortex or just of other different species. But what I'd also like to show in the next part, the last part of this talk, is how we're using these approaches not just to expand on our ability to molecularly profile the human cortex, but also our ability to perturb and observe different hypotheses in these in vitro models. And specifically here, what I'm alluding to is our ability to generate now high throughput perturbational screens of the human cortex in different models to ask how do not just like different cell types form, but how indeed our whole cortical areas form in the human brain. Now, this is particularly important because actually virtually all of these neurodevelopmental disorders manifest as some sort of phenotype localized to a specific cortical area, whether that be altered neuronal activities deep inside the brain or molecular changes that have been profiled in the front of the brain in your prefrontal cortex. And many of these profiles also show actually that in patients with neurodevelopmental disorders, the molecular distinctions between these different cortical areas are dampened. And so to ask what is the link between the emergence of these neurodevelopmental disorders and the formation of these cortical areas, I developed a pooled CRISPR activation screen to first ask what are the transcription factors that are driving areal identity to begin with. And so these screens exist, or we have conducted these screens in human primary cortical tissue. And I first started by asking what are the transcription factors that drive the specification of prefrontal cortex fate, or PFC fate, because this area has already been so extensively molecularly profiled. So for each of the tissues in my screen, I dissociate them, I plate them, I infect them with a guide RNA library against PFC targeted or PFC transcription factors, as well as a calculated Cas9 tagged to an activator domain. After 10 days in culture, these cells still retain relatively healthy morphology. And so using flow cytometry and single-cell RNA-seq, I can then profile these perturbed cells. And what you'll see here in the UMAP of this perturbed data set is that these cells still retain the cell types that I would expect in a human cortex. Okay. So now with these perturbed cells in hand, I've developed a bioinformatic pipeline to ask what are the guide RNAs and therefore what are the perturbation targets in each of these cells and how do those perturbations make them closer to our reference data set, that is a single cell transcriptomic data set of the developing human primary prefrontal cortex. And so by taking these perturbed cells and then mapping their transcriptome to our reference, we're able to find several interesting hits, including this transcription factor called YBX1, where you can see that with, to an extent, increasing levels of YBX1, we see a similarity to this reference PFC or reference prefrontal cortex, but too much YBX1 actually decreases the similarity to the PFC in a sort of witch hat fashion. Now, teasing apart what's underlying this witch hat pattern is going to be difficult in human primary cortical tissues because of the relative inaccessibility of this model. So to make sure that I can actually have a mechanistic platform to test what YBX1 and other hits are doing, I wanted to take this screen and now apply it to our organoid and chimeroid model. So I took the same guide RNA library, I put it into stem cells, I made the stem cells into organoids, and then using that same reaggregation and infection approach I showed you earlier for the meta-atlas paper, I put DCAS9A into these organoids and grew them up, and then profiled what happened to these perturbed cells. Now, what's exciting here is that all of these perturbations can still produce organoids with that classic rosette structure lined with mature neurons that you would expect from a healthy cortical organoid. And even more excitingly, the hits that I just showed you, particularly the hit with YBX1 from the primary tissue screens, that is still retained in our cortical organoid model. So this suggests that we now have this cortical organoid platform that we can use to have targeted mechanistic analyses of these determinants of areal identity. And so ongoing right now, I'm analyzing work where we ask, is YBX1 even required for PFC fate? And here we're using the same platform of shRNA knockdown analyses that I showed you earlier to ask how do FESF2 and TSH3 drive deep layer fate. But we can also use that CRISPR activation platform to ask, how does YBX1 and other PFC determinants, how do they play with determinants of other areas? For example, in the back of your brain in the visual cortex, the field has established that a transcription factor called NR2F1 is really required for specifying visual cortex fate. So if you have YBX1 and NR2F1 overactivated, which of these areas wins? I'm very excited to share the results of this analysis whenever we have it done in a month or so. But for now, I'd like to end and summarize that we've shown you approaches both from our paper and from our ongoing work of how we can create massive, more comprehensive molecular profiles of the developing human cortex, test and then take up hypotheses from these profiles and test them in vitro models. And with an eye towards the future, I'm excited to take these types of technologies and ask questions about now, how do neurodevelopmental phenotypes form? What I imagine here are experiments such as taking that perturbational platform and now asking how do risk genes for certain neurodevelopmental disorders drive the formation of area-specific molecular signatures. I also see a world where we can take these perturbational approaches and couple them with readouts or fluorescent readouts of electrophysiological activity, now asking how these risk genes affect not just area-specific molecular signatures, but also area-specific neuronal activities. And you can also imagine taking these approaches and looking at this question from the opposite view, deriving patient-derived cortical organoids and using my bioinformatic pipeline to ask not just what are the developmental trajectories in these patient organoids, but also can we modulate any of these area-specific transcription factors to course correct any disrupted development. And with that, thank you again for this chance to speak. I'm excited for a discussion, but first I'd love to thank my very supportive mentor, Dr. Aparna Bhaduri, our highly collaborative lab, our collaborators, and all of you for your attention. Thanks again.
Thank you so much. That was a really great presentation and overview of your recent work. If anybody has questions, feel free to put them in the chat, or you can maybe raise your hand. I'm not sure how it works on this, but feel free to add them to the chat for sure. Maybe while we're waiting for those to come in, I can ask a first question. When you are using these chimeroid models, how do you think about controlling for the non-cell autonomous signaling from a knockout neuron next to a differently affected progenitor or whatnot, signaling to each other as they're developing?
Yeah, that is a great question. I'm trying to pull up this. Oh, here we go. So, these transcriptomic readouts, they don't give us spatially how are a perturbed cell next, like interacting with a non-perturbed cell. I've tried to account for that with several controls. So you'll notice here that everything I'm doing is related to in a specific organoid, a perturbed cell versus a cell that doesn't have a perturbation. And we also have non-targeting or scramble controls as well. But I do think that to really truly address this question, I'm really excited to start interplaying with spatial transcriptomic data or either data sets or using that as a readout with these types of perturbations.
Thanks. I see a question here. Very interesting talk. I got in a bit late, and I'm wondering if you could explain a bit more how you went from gene programs to nominating a transcription factor to go after in vitro studies.
Yeah, so really how that part of the talk happened is that we have this module 20, and what we found just by looking at this module is expressed in other data sets and interacting specifically here with an adult cortical data set is that this module seems to be most active in deep layer subtypes, but those are specifically elevated with this FESF2 transcription factor. But that was weird because this FESF2 transcription factor, the classic driver of deep layer fates, it's not actually present in module 20. And by interacting with previous literature, including work from Palarlada's lab that has looked at putative FESF2 targets, that's what led us to the fact that, oh, one of these putative FESF2 targets is actually the only transcription factor in module 20. And that's kind of the line of thinking that we saw to get from modules all the way to individual transcription factors.
Thank you. Are there any other questions?
Can I ask one question here? So I think you use a lot of like published data, for this kind of analysis. I was just wondering when you try to use the single cell data from the published paper, do you use the FASTQ files and do everything from scratch, or do you actually basically use the annotated single cell data for the analysis? I mean, cell type annotation is kind of time consuming, and also, sometimes, we actually use published data, but when we try to redo the annotation, the annotation is different, right? So I was just wondering, how do you actually handle that?
Great question. So we don't start with FASTQ. We start from the counts matrices. And then for, I believe, most of them, we apply the quality control metrics that they suggest. But then what we do with these count matrices, we generate a Seurat objects for each individual. And then to make that UMAP that I keep showing, we can do conventional batch integration techniques to put all of those individuals together. We actually make our own cell type annotation from that integrated UMAP data set because, of course, of the same types of limitations that you just talked about. But I just want to add on to that's what the strength of the meta-module approach is for. You can imagine that there are also limitations with conducting cell type annotation on integrated data. The meta-module generation, since it starts from each individual without having to integrate them together, and as you can see, since some of these meta-modules are actually linked to certain cell types, they provide us with an orthogonal approach that can allow you to annotate this data independently of batch integration.
And I think that this is kind of an important point as these data sets come out. I think that this is one thing that we were pleasantly surprised by, and we wanted to try with looking at the counts level matrices because it just changes the throughput of how much data you can put together. And there's so much technical artifact. And kind of how we were thinking about this is also when you're dealing with human samples, especially developmental samples, there's so much, well, actually in general, I think it gets worse as you get into adult. There's just so much you can't control about about the process, right? You can't change anything about the clinical collection. You can't change anything about what the time period is between collection and acquiring the sample. You just have to go with what the collection resources and whatever is convenient for them. And that's totally understandable and correct. But then it winds up with a lot of individual to individual variation. And so if you look at a lot of individual data sets from either development, but also different disorders, or we do a lot with cancer, you see the same thing where there's patient-specific batch effect. And that's just going to be compounded by the techniques, the analysis, the alignment, which genome is used for those alignments. And so we really wanted to see, and this was kind of a shot in the dark, was if you just go one level above that, can you find biological variation that is preserved across everything? And we agree that you're probably losing some of the detail, but I think what Patricia saw and what we were really excited about is there's so much low-hanging fruit in terms of mechanism that's not understood, that perhaps one day in like 15 years, when we're done doing all of the experiments for the things in the hypotheses we can develop now, we'll have to become more granular and find some of those more subtle differences. But for now, it seems like the trends that we're finding are also cross-applicable to different data sets, different individuals. And so they seem to be deeper principles and stronger phenotypes. And so we're really excited about that. And I think that that really gives us confidence as we move into more of the patient world of thinking about patient-derived samples from neurodevelopmental disorders and really benchmarking them against some of these data sets, et cetera. So I think it was really exciting for us, but it wasn't something that we knew the answer to when we started. But we're hoping that this can be a resource for the field for other people to also look at that kind of higher level and just be more empowered to use these data sets.
Thank you so much.
Right, there's another question in the chat here. If I understand correctly, did you run the gene program analysis independently for each individual and then combine them to check which programs overlap across individuals?
So what I did here was the first step of the metamodule pipeline has to be done per individual. And that first step is conducting hierarchical clustering within each individual to get what are the biological variation, what are the signatures within those samples. It's not necessarily asking for what are the overlapping cluster markers between these different samples. Instead, what we did is that we then curated what are the genes that are the most representative of the cluster markers within these different samples. And then we hierarchically clustered those to generate these metamodules. And we clustered them, I remember correctly, by how that gene score metric, by how specific and enriched they are to different markers. And so it's not really just a Venn diagram of what are the individual cluster markers, it's really asking how do they travel together throughout the entire space of the meta-atlas.
Thanks. Any other questions from the audience? I might ask one more that I had here. So you mentioned at the end how you're using this platform that you've developed to understand the role of ASD genes and how different transcription factors you've identified might rescue some of these ASD genetic variants. Do you think that your platform is also useful for identifying transcription factors that might have a role in generating more specific subtypes of iPS-derived neurons and model systems for studying those disorders?
Yes, I think so. So what I can see here, and then let me know if this answers your question, is a world where from these different screens, so for example, from this PFC screen that I was talking about, I see a world where we can get neuronal subtypes that maybe we didn't see in cortical organoids before, but more fully capture and mimic subtypes that maybe we see in our developmental meta-atlas or in other primary tissue data sets. And then what it can tell us then is like, the overexpression of this transcription factor, it seems to really drive the specification of this specific neuronal fate. Similar type of thinking as what we saw earlier with FSF2 and TSHD3. And then I think taking that type of finding and then continuing to dig deeper, maybe we have to overexpress it in a certain promoter, or maybe we had to combine it with the overexpression of a different transcription factor, then we can get this subtype that we're really seeking for. I can see the platform I'm going for or that I'm developing helping towards that lens.
Thanks. There's lots of potential. All right, if there's no other questions, oh, we have one more, Yixing. Go ahead, you have your hand raised. I think you can.
Yes, I just have a technical question. I noticed from your method in the paper, so for the developmental and other atlases, so it seems that you actually use different like serial version, maybe even a pipeline to integrate the data, like version 4 and 5. I'm just wondering whether it's just some historical reason behind that, or like if you are going to redo it for now, so would you prefer, or which pipeline would you prefer?
Oh, this is actually a very good question. So the biggest reason why the adult meta-atlas is integrated not in a slightly different way is because it's simply too big. It was very difficult to integrate 2.6 million cells as opposed to 600,000 cells for the developmental meta-atlas. And at that point, the sketch integration pipeline from Seurat had come out, and so we were able to mix them together. I think if there's concern about what these different integration techniques actually do to the data, it's very valid. That's why we're excited about the meta module approach, because it gives that orthogonal, integration independent annotation of all these different cells.
All right. Thank you, everybody, for joining today. And please join me in thanking both of our speakers, Dr. Bhaduri and Dr. Nano. Thank you so much for your presentations today.
Thank you so much for the invitation. And if anyone has follow-up questions, please don't hesitate to email us. Thank you.